Welcome to this explainer. Okay, so as med and grad students, you already know breathing is non-negotiable, but today we're getting into the clinical math behind it. I mean, think about it. A healthy adult takes roughly 20,000 breaths every single day. It's this automatic rhythm we totally take for granted, but we're going to trace the wild, kind of treacherous journey of a single oxygen molecule. We'll follow it from the outside air all the way down into the lungs and across a microscopic liquid interface right into the blood. It's a journey governed completely by biophysics, where one tiny microscopic glitch can cause a patient to just crash. Understanding the math here, that's literally how you're going to diagnose and treat respiratory failure in the wards. So here's our roadmap for today. We're going to hit the alveolar capillary interface, decode Ficks law, look at maximizing diffusion, examine tissue thickness barriers, and finally wrap up with what happens when this whole system fails. All right, jumping right into section 1. The alveolar capillary interface. Let's look at exactly where the magic of gas exchange happens. To really grasp pulmonary diffusion, we've got to look at the ultimate end goal, right? Oxygen comes in from the atmosphere, travels down the bronchial tree, and fills up the alveoli. But here's the kicker. The air and the blood don't actually touch. There's this incredibly critical microscopic gap between them, a liquid interface. This isn't just empty space. An oxygen molecule has to literally dissolve into a layer of pulmonary surfactant, pass right through the alveolar epithelial cell, cross a basement membrane, swim through interstitial fluid, and finally pierce the endothelial layer of the capillary just to hitch a ride on a red blood cell. From there, it's off to the tissues to synthesize ATP. This multi-layered liquid gauntlet is a massive physiological bottleneck. If diffusion across this space drags, your tissue starts screaming for oxygen immediately. And navigating that bottleneck brings us right to section 2, decoding a Ficks law. Let's look at the strict mathematical rules of the game. Okay, let's dive into this. Ficks law of diffusion is the foundational biophysical law that explains exactly how we get that oxygen onto the red blood cell. It states that the diffusion, or the actual flux of gas across an interface, is determined by a very specific formula. Basically, flux is directly proportional to a set of driving factors on the top of the equation, the numerator, and inversely proportional to the physical barrier on the bottom, the denominator. And I really want to emphasize something here. This is not just some abstract equation to cram for your boards. No way. This is the literal, mathematical difference between your patient breathing comfortably on room air and your patient coding in the ICU. So let's break that equation apart. Moving to section 3, maximizing diffusion, or as I like to call it, the numerator. There are 3 primary variables in the top half of Ficks' equation that really push and promote the flux of gas. We've got the diffusion coefficient, the surface area, and the concentration gradient. When these 3 variables are firing on all cylinders, oxygenation is highly efficient. Let's look at each one to see exactly why. First up is the diffusion coefficient, or D. This is basically just a measure of how easily a specific gas permeates biological membranes. Now here's a totally fascinating clinical pearl. This coefficient is a constant that is unique to each individual gas. For instance, the carbon dioxide produced in your tissues naturally has a way higher diffusion coefficient than oxygen. It actually diffuses about 20 times faster. That is huge. Because CO2 slips across these membranes so effortlessly compared to O2, a patient with a diffusion impairment is almost always going to become dangerously hypoxic way before they become hypercapnic. Basically, the oxygen always fails first, just because of this single coefficient. Next up is A, the surface area available for gas exchange. Get this, in a normal human lung, we have roughly 500 million alveoli, and pretty much all of them have at least one capillary perfusing them. To put the sheer scale of that into perspective, if you were to unfold and flatten out the internal surface area of all 500 million of those little sacs, it would cover an entire tennis court. Your body literally packed a whole tennis court's worth of surface area right inside your chest, just to maximize that numerator in Ficks' law. It's kind of wild to think about. Now, let's see how this actually builds a clinical picture. Compare that normal, massive surface area to a diseased state, like emphysema. In emphysema, chronic inflammation actually destroys these delicate little lung units. The alveolar walls break down, they merge together, and suddenly, that huge tennis court-sized surface area just plummets. Clinically, you're going to see a patient with hyperinflated lungs, usually presenting with a classic barrel chest. They're physically struggling, trying to pull in enough oxygen to make up for that lost area. Because surface area lives in the numerator of Ficks' law, when it goes down, the overall diffusion and flux of oxygen into the blood crashes right along with it. The final piece of our numerator is the concentration gradient. That's the partial pressure of oxygen in the alveoli, which is P1, minus the partial pressure of oxygen in the blood, or P2. You remember the alveolar gas equation, right? Well, that determines your P1. Gases naturally want to flow down their gradient, from high pressure to low pressure. So the more oxygen you have in a well-ventilated alveolus and the less oxygen on those returning venous red blood cells, the steeper the gradient. And a steep gradient forcefully drives oxygen into the blood. But if a patient can't ventilate properly, that pressure gradient is ruined, and diffusion literally stalls out. Definitely keep this variable in your back pocket, because manipulating this specific gradient is one of our absolute best tools in emergency medicine. All right, moving below the fraction line to section 4, the barrier or tissue thickness. This is the denominator of Ficks' equation. So that entire numerator we just talked about, it gets divided by the physical thickness of the interface the gas actually has to cross. And this brilliantly illustrates the delicate balance of the lung. In a healthy state, this barrier is mind-blowingly thin. We're talking roughly 0.6 micrometers. Because it's so thin, gas flux happens in a flash. But any pathological increase in this thickness immediately tanks diffusion. Since we're dealing with the denominator here, as that physical wall gets thicker, the total oxygen flux gets smaller. So what does a messed up denominator actually look like in your patient? Let's look at two classic examples. In pulmonary fibrosis, you get this abnormal deposition of extracellular matrix. The tissue around the exchange units actively scars and thickens, building a literal, tough physical wall that makes it super hard for oxygen to cross. Another incredibly common one? Left-sided heart failure. When that left ventricle fails, hydrostatic pressure backs up straight into the pulmonary circulation. That high pressure literally shoves fluid out of the capillaries, causing cardiogenic pulmonary edema. You end up with water pooling right between the alveolus and the capillary. This extra fluid pumps up the thickness of the barrier and actively suffocates the diffusion process. Bringing it all together now in section 5, when pulmonary diffusion fails, let's look at the overall impact on our patients. To really appreciate the pathology here, we have to respect the normal physiology. In a healthy, non-fibrotic lung, with that huge surface area and perfect gas gradients, diffusion is breathtakingly fast. Think about this. The normal transit time for a red blood cell zipping through a pulmonary capillary at rest is just about 0.75 seconds. But because all those Fick variables are so perfectly optimized, that red blood cell hits complete oxygen equilibrium in only 0.25 seconds. It is fully saturated in well under a single second. That's just how perfectly dialed in Fick's law is in a healthy human body. Now, the whole point of this is learning how to map real diseases directly to this biophysical math. Check out this table. Look at emphysema. It actively attacks the numerator by wiping out surface area, which plummets total gas flux. Then you've got pulmonary fibrosis and left-sided heart failure. Both of these go right after the denominator. One builds a rigid wall of scar tissue, and the other builds a boggy wall of water. But the mechanical result is exactly the same, an increased barrier thickness that drastically cuts down gas flux. Pretty much every respiratory disease you'll ever see on the wards is manipulating this exact equation. I want to leave you with a quick clinical puzzle to really test your grasp of this math. Picture this. You have a hypoxic patient crashing in the ER right in front of you. You know Fick's law is failing them. If your absolute top priority is to immediately increase that oxygen flux, which specific variable in Fick's equation is the absolute easiest and fastest for you to manipulate therapeutically? Think about the numerator. Think about the denominator. What can you directly control the very second you step foot in that room? I hope this explainer has shed some light on the beautiful, vital math of pulmonary diffusion. Keep analyzing, keep questioning, and I'll catch you next time.